Hi everyone, in this video I'm gonna show you how to automatically count cells and measure their area using ImageJ. First, I open ImageJ and load my image. So I go to image type. You could choose 8-bit or 16-bit. By the way, this is DAPI staining of cells, so you want to count the stained cells, the living cells. Then I go to image, adjust and threshold. Take a moment to check that small noise in the background is not selected and that the cells are not merged into one big blob. In terms of threshold, you could click on red, black and white or anything else, but the important point is you should select the best possible area. Remember, for a series of samples, if you want to compare them, as far as possible, use the same variables, threshold or other choices. Try auto, it usually gets better results. Then click apply. If your cells are stuck together, they may still be counted as one object. To try to split them, I go to process, then binary and click on watershed. I go to analyze particles. This window controls how ImageJ finds and measures each cell. In size, I can set a minimum area to ignore tiny noise. You can also consider a minimum circularity to increase the accuracy, because almost all cells have a circular shape. You can exclude the cells on the edges of the image or not, but if you choose to exclude or include, do that for your all samples. Click on pixel units. So you can see your results and now your cells are counted. The important information are in the summary sheet. You can see your cell count, total area, average size and percentage area. Thanks for watching.